Benthic macroinvertebrates are small organisms that live at the bottom of rivers and lakes, where they can burrow into the ground, controlling the flow of nutrients in the ecosystem. They serve as decomposers and food for larger animals such as birds, turtles, and fish. They can also indicate the quality of water. If there is high species richness, then the water probably is clean according to the EPA standards. Some examples of macroinvertebrates include crayfish, insect larvae, worms, and gastropods like snails. These small animals are commonly found in riffles, which are geographic structures that lie on the bottom of riverbeds and cause turbulence in the flowing water, which also provides shelter from predators. The strength and size of riffles are determined by the size of the rocks on the riverbed. The larger the rocks, the larger the riffles, which means more oxygen and nutrients can flow into those areas. In one study, a group created different sized artificial rocks to increase the productive capacity of a stream, which leads us into our own hypothesis. An area with bigger rocks in the river contains a higher population size than an area with smaller rocks because benthic macroinvertebrates prefer riffles that the bigger rocks provide. The first step of data collection is to locate the best riffle that would provide us with the most suitable representation of the populations present in the North River. Following the first step, we gathered other important data such as stream width, depth, flow rate, and temperature as seen in the following clips. We used two different methods to find stream width using both a measuring tape and a rangefinder. To measure flow rate, we used the orange method, calculating the flow based on the speed at which the orange traveled a certain distance. Lastly, gathering depth with a meter stick and temperature with a thermometer. Once those steps are completed, it was then time to secure the kick net at the bottom of the riffle. Rocks are then taken from outside the sample area to be placed along the bottom of the net to tightly secure it along the bottom of the river. This ensures less chance of organisms avoiding collection. With the net secured, we begin rubbing the rocks thoroughly to make sure that any macroinvertebrates that live on the rocks are removed. Following immediately after this, the bottom is scraped vigorously for the remaining amount of time to gather the organisms that live on the bottom of the river. Lastly, the kick net is removed from the water and taken to the table for the organisms to be removed. Once removed, they are then organized and identified. During the collection, random rocks were gathered from the sample area to be measured. Chung states in his research that larger rocks will allow for larger riffles, which allow for more flow and distribution of resources of which benthic macroinvertebrates can benefit from.
We calculated the average volume of rocks from each data collection set and compared it to the number of species present. The average rock size did not have much of an impact on the number of species we captured and shows no real relationship. The highest number of species was 11 on the first day, and the lowest number of species was 5 on the last day. We also compared the rock size with the number of individuals present. The rock size did not have much of an impact on the number of individuals we captured, and it also shows no real relationship. The highest number of individuals was 148 on the first day, and the lowest number of individuals was 17 on the last day. Day 3 had an average volume of 1,233 cubic centimeters that could be seen as an outlier on both rock size graphs. However, we were able to gather from our data that there was a positive correlation between species diversity and water temperature. The highest number of species collected was with a temperature of 14.3 degrees Celsius on the first day, and the lowest number of species collected was with a temperature of 9 degrees Celsius on the last day. We also compared the water temperature from each day to the number of individuals present. The water temperature shows a positive relationship with the population density. The highest and lowest number of individuals collected were with the same temperatures from the species diversity and water temperature graph. According to a study on the stream succession done in Glacier National Park, macroinvertebrate taxon richness and the cumulative number of taxa over the study pyramid displayed a strong positive relationship with water temperature. Day 2 had a water temperature of 9 degrees Celsius and could be seen as an outlier in both the water temperature graphs because it shows a high number of species and individuals present, while Day 6, which also had a water temperature of 9 degrees Celsius, had a low number of species and individuals present.
In conclusion, based on our findings, we failed to reject our original hypothesis that larger rocks affect the population density of benthic macroinvertebrates. This research was significant because there isn't a relationship between rock size and the amount of species found. So smaller streams with a lot of small rocks can be as diverse or even more diverse than large streams with a lot of large rocks. Well, there you have it. Thanks for watching. Signing off.